of Italy. And I was particularly interested in how plants support agriculture. And so for this reason, I decided to enroll in biotechnology applied to agriculture at the University of Bari, which is a local university in the south of Italy, of my region. And there I also fell for plant genetics, because I saw it as like an instrument to understand like what define a specific plant organism, but also it kind of allows us to study its history in terms of parental relationship, but also evolution. So afterward, I decided to enroll in graduate school. I got a PhD fellowship at Fondazione Mudmac, which is a research center in the Alps, which works mostly on apple and grapevine. They are the main crops in this area. And my PhD was in collaboration with University of Padua, and focused mostly on grapevine genomics. And so the Fondazione Mudmac, besides being a research institution, has also provided and advising services for growers. And so there I understood that I was very interested in how our knowledge is kind of an interface between basic research and applied, and I got very interested in plant breeding. And so then I decided to accept a postdoctoral position at University of California, Davis, where I work within the Walnut breeding program. And afterward, I arrived here in the cold Massachusetts, and I'm working with Brooks. And here we are trying to understand how we can use plant breeding and plant genetics approaches to understand rice adaptation to drought. So if I have to summarize the three main topics that have kind of inspired me so far, but they keep interesting me, I can say I summarize in three questions. How did agriculture affect crop diversity? How does plant breeding support current agriculture, and how we can use the modern technology to adapt agriculture to the future? And I'm gonna give some example in grapevine, walnut, and rice. So I will start talking about my PhD thesis, where I was interested in understanding the phenotypic and genetic diversity between the wild and cultivated grapevine. So when I talk about grapevine, I'm referring to the Eurasian species Vitis vinifera, which we can find today in two forms. The first one is the wild form, Vitis vinifera sylvestris, and the second one is the cultivated form, Vitis vinifera sativa. It is supposed that the cultivated form was domesticated from sylvestris about 8,000 years before present in the Near East, even if there are theories that multiple domestication centers have occurred across the year. So these two subspecies present many morphological differences, such as flower sacs, leaf, bunch and berry size, and seed shape. For instance, the wild form is dioecious, meaning that it carries the male and female flower in two different plants. And in general, the berries are very small, very acid, and they have a very thin, fleshy flesh tissue. Today, the wild form is presenting small and isolated population in Eurasia, and Italy and Spain, they are both the highest number of wild population. On the contrary, the cultivated form is the one that we are more familiar with because it is the one used for wine production, but also as table grape. So the cultivated form is hermaphrodite, so you have both male and female flowers on the same plant, and it's very variable, so you have a variability of shape and color of the berries depending on the use. For instance, table grape in general have bigger berries compared to the wild varieties. So this diversity in phenotype is supported by a high level of heterozygosity. In general, it's like on average 0.3, and this is the results of three main evolutionary forces, which are sexual reproduction, and vegetative propagation and somatic mutation. So vegetative propagation is the main agronomical practice in viticulture, And it kind of preserved the genetic profile of an individual. And this is very important when we want to produce wine very authentic, like Pinot Noir and so on. However, recent studies have shown that while we are fixing the genetic profile that we care, there has been also a fixation of recessive deleterious mutation in an heterozygous status, which means that most likely this individual could have less fitness in the future. So this is why in the last decade there's been an interest in studying the Silvestris. So the main objective of my thesis were to understand which is the current status of genetic diversity in the wild population. And also we want to see if the phenotypic divergence of these two subspecies was also supported by a genetic differentiation genome-wide. So we worked with a collection of about 100 of individuals, which were collected at our local grapevine germplasm repository. This repository has a very high number of authentic Silvestris. And why I wanted to highlight the authenticity of the material is that sometimes it's very difficult to be sure that you are working with authentic wild population because you can confuse a Silvestris with a feral sativa, which are basically cultivars that have just been abandoned in the field. We were sure because, first of all, these individuals were accurately analyzed using morphological parameters, but also with an accurate molecular analysis. So we genotyped this population using two approaches. The first one, we apply the 20K grapevine Illumina SNP array, but also we set up a new method of restriction site associated DNA sequencing. So we started with about 54K SNPs, but then we applied quality filters and we end up with about 27K SNPs. So we use these genetic markers to understand how the two subspecies are, how much they are diverse. So as we were expecting, they are divided in two subgroups. And I say expected because they were already sampled after a molecular analysis. So the SNPs, they kind of confirm the analysis based on SSAR markers. But here you can see already that the cultivars are very diverse compared to the Silvestris, which look more similar to each other. And these results were supported also by the genetic diversity metric. You see that in the cultivated grapevine, there is more heterozygosity compared to the Silvestris, where we see a higher value of inbreeding coefficient. And this is quite rare because in many species, you are expecting to have more diversity in the wild population than the cultivated. But for three species, this is not always so obvious. And so these results reflected also the linkage disequilibrium decay. So overall, the LAD decay within 10 kilobases in the whole population. But if we consider the wild population Silvestris, the decay was lower. The R-squared reached value below 0.2 in 20 kilobases. So these results altogether suggested that the genetic diversity of Silvestris is lower than the one of the cultivated grapevine. So most likely the Silvestris has a small effective population size. But we have also to say that most of our material was Italian, so it could also be that this situation is related with the Italian wild population. On the contrary, the sativa population is a big meta population with overlap generation. And as I say, this is results of crossing, somatic mutation, and massive vegetative propagation. The genetic differentiation estimated using the right coefficient was overall moderate. And this is because the two species are interfertile, so there can be gene flow between the two. However, when we look at the FSAT value, across the genome, we saw that there were different genomic regions that were highly differentiated between the two subspecies. And at this genomic region, the Silvestris had, in general, lower nucleotide diversity. So we look at which kind of genes are in this genomic region, if there is kind of enrichment of genetic function, of gene function. So first of all, we saw a signature selection here on chromosome 2, where previously studies have mapped the sex locus. So this was kind of a confirmation that our analysis worked well. But also we found gene related with leaf morphology that, as I recall, is one of the differences between the two subspecies. We also find gene related with berry size and composition. For example, the nucleotide transcription factor 2 was within a signature selection chromosome 17 that previous work have correlated with berry size, and they have, and it is thought that he's like a domestication locus. But we saw a particular enrichment in stress-related genes, a result that has been also confirmed afterwards using whole genome sequencing from other groups. So these results kind of are telling us that while the grapevine that is cultivated in the vineyard is completely dependent on human action, in the wild population, they keep facing the environmental changes. And also, like, there has been a huge decline of wild individuals due to human urbanization, but also the introduction of pathogens from America, which were kind of resolved by introducing American species in the late 19th century in the vineyard, but not in the wild, in the natural habitat. So we suspect that the Silvestris can be, the Silvestri that we see today, they can be potential donor of resilient allele to use in viticulture. And this is why from that moment, many groups are now trying to understand if this is true, and they are trying to see which kind of resistance and tolerant gene are in this subspecies. So as I said before, during this year, I see that, so it was very interesting to understand. I got like interested in evolution and so on, but I was also asking myself, okay, well, how we can actually use these results to improve agriculture. And so then I moved to the United States, and I started working within the California Walnut improvement breeding at UC Davis. So United States is the leading country for walnut export, and California accounts for more than 99% of the domestic walnut production. But also in California, there is one of the most important breeding program of walnut worldwide, and is at UC Davis. So the UC Davis WIP was established in the late 1940s, and since then has worked to address growers' needs in California, and has released many varieties, including Chandler, is a very famous and popular walnut cultivars, and is probably the one that you will find on the shelf of your grocery store. So, but how they did that? So far, the breeders were basically walking in the orchards. They were looking at the tree and measuring traits and selecting the candidate parents based on how they looked. But this phenotypic selection can be inaccurate because, as we know, phenotypes depends on both genotype and environment. So you don't know which one is actually influencing the trait in that moment. And also, like, phenotypic selection is laborious and time consuming. And this is even true when we are talking about tree. For example, in walnut, if you have to see if your selection worked good, you have to wait at least 9 years till you have a stable producing tree. So for all these reasons, it is important that for a breeding program as the one at UC Davis, we introduce marker-assisted selection, which is basically the possibility of selecting an individual on its DNA information. So it is more accurate, and also it allows you to select at the seedling stage, so when the plant is very small, and to select for multiple traits. So for this reason, the California Walnut Board decided to find a 5-year project with the aim of developing genomic resources for walnut, and the final goal of applying this technology to identify markers that then can be used to select and develop promising walnut varieties. So this roadmap started with release of the reference genome. The first version was released in 2016 before my arrival, but this version was very fragmented in small contexts. So we decided to improve it by applying third-generation sequencing approaches. In particular, we use the Oxford Nanopore Mini-ion combined with the long-read scaffolding technology IC provided by Dovetail. We also use isoform sequencing, which is a technology provided by PacBio for sequencing long transcripts, and is very useful for improving gene annotation. So here you can see that the new version of the genome is very contiguous, and we were able to assemble the 16 chromosome pseudomolecule. We also predicted 37K gene models that have a longer sequence compared to the previous version on average. But also we reduce redundancy. What I mean when I say that, so walnut is a very heterozygous species, which means that sometimes you can have that a specific region has different variation in sequence, different variant. And you can, if you don't have a good technology, you can be mistaken and annotate these two variants as two different scaffolds. So we saw that in the first version, there was a lot of redundancy, and thanks to the long-read sequencing approaches, we kind of removed them, improving a lot the accuracy of the genome. But also we were interested in the level of heterozygosity of Chandler across the genome. So here in this heat map, in blue, you have the region that are particularly heterozygous, and in white, you have the region that have low heterozygosity. So besides the region that they correspond to the centromeres, we saw that there are some genomic regions in Chandler that are particularly wide. So they have a particular deficit of heterozygous loci. We were particularly interested on chromosome 15 because it is the shortest chromosomes, but also has extended region of high homozygosity. So we suspected that one of the possible reasons of this low level of heterozygosity across the genome could be inbreeding. So we look at the pedigree of Chandler, and so here you have the pedigree, and you see that both the father and the mother of Chandler have a common ancestor, which is Pain. Pain is a variety that was introduced at the beginning of the walnut breeding program to improve yield, and so has been extensively used in the breeding program. And so we say, oh, probably the presence of pain in both lineage can be the cause of this high level of homozygosity. So here you have chromosome 15, and in the inner cycle you have the green, in green the region that are homozygous, and in gray you have region that are heterozygous. And you see in the outer circles, they indicate the maternal and the paternal alleles. So you see that in correspondence of the green region, you have the same allele heredity, the same alleles in both mother and father. And these alleles are the same of Pain here in orange. So this kind of allowed us to say, oh yes, there are some region of Chandler they have inbreeding. And this is important in walnut because it's a species that suffer of inbreeding depression, a phenomenon that can cause some problems such as sterility. And so this was possible thanks to the development of the genome. And so, and for sure will help the breeders to be more accurate in the future crossing. But in this trend, we also say, okay, after we develop a genome, we have also to assess the entire plants in the breeding program and how we do that. So we developed a SNP array, which actually include over 609,000 SNPs. And we use this tool to genotype over 1,200 accessions of the breeding program. So this chip had many applications, but one that is very important is the pedigree reconstruction. So the pedigree is like kind of a Bible in a breeding program because allow the breeders to select which individual to cross and how to manage the different crosses across the years. And so it has to be accurate and robust. We use the genotype information at the SNPs to reconstruct the pedigree. And so how we did that? We estimated for each pairwise comparison the coefficient of relatedness, which is this kinship coefficient. And here on the x-axis, you have the identical by state 0, which indicate the proportion of alleles that are different between two individuals. So here are the dots, each of them represent one of the pairwise comparison, and the color indicate the level of relationship between individuals. So in blue, you have the parent offspring. In orange, you have the full sibs, in green, the half sibs, and in purple, the unknown. So thanks to the SNP, we were able to confirm most of the relationship that they were registered in the historical pedigree, but also we discovered new parent-offspring relationship. And also we identified some errors, basically duplicates, that they were called with different name, but they were the same individual. And these are errors that usually occurs in the breeding program because during different phases. So thanks to this SNP array, we provided a robust pedigree to the walnut breeding program. And so now the breeders can use it to avoid inbreeding depression and also to use at the best the genetic variability available. But the last objective was, of course, identifying markers that we can use to select the best promising individual. And in the breeding program at UC Davis, they are particularly interested in 5 traits. 2 of them are related with the adaptability of an individual to specific geographical condition. So it is leafing date, which is the time at which more than 50% of the leaf are out. And in California, they aim to have late leafing individuals, because in this way you can skip some risk of walnut blight, which is a serious disease for in California, and also like some risk of late spring frost. Harvesting date is the time at which the husk, which is the green tissue outside, break, and you can harvest the walnut. And in California, they are interested in having early harvesting individual because so they have a spread season to harvest walnuts. Then of course they are interested in improving yield. And here you have the fruit bearing, which is the ability of an individual to produce lateral fruiting buds. And of course they select for lateral individual because they produce more walnuts. And this is a consumer preference trait, which is kernel color. So consumers seems to prefer light kernels, and this really surprised me because in Europe we prefer this kind of walnuts, but it was very an interesting story. So we identify market rates for all of them for the first time, but I'm not gonna give all the information on that. I'm just gonna give an example in leafing date. Because I got very interested in phenology, because this phenology is the kind of, so trees are reacting to the variation in climates that we are facing by changing their phenological phases. For example, in Walnut, we are seeing that in some region leafing date is anticipated because winters are warmer than before, but in other region it is delayed because walnut requires chilling during the winter. And so probably in this region, the chilling requirement is not satisfied and the tree is very lazy to wake up. And this can have serious issue because you can have damage of frost, and also the harvesting season is delayed, and this is a huge problem, especially for local growers. And so we identify a major locus on chromosome 1 for leafing date. And here you have one of the SNPs that mostly explain the variation of leafing date. So here in 1 you have the major allele, and in 2 you have the minor allele. And you can see that when an individual has the 2 allele, there is a delay in leafing date. So this markers is promising to use it as to select late leafing individual, and is already being validated in the breeding program and probably will be used in the future. But the takeout message of this part of the talk is that we were able to develop many genomic resources in the Walnut breeding program that are useful not only in California, but also in other parts of the world. For example, they have been used in France and Iran to study other traits, including water use efficiency. So with this project, we kind of provided a bunch of tools that can be useful for improving agriculture and improving walnut production. And then I'm at the last part of my talk. While I was at the breeding program, I had many experience in talking with growers, and I saw that many times we are worried about the present, like we are worried about the introit that we can have now, but we are not worried about what it can happen in the future. And so I'm particularly interested in how agriculture will be affected by the climate change that we are facing. So here at UMass Boston, I'm studying rice adaptation to drought. And here today, I'm gonna talk about some preliminary results that we have about the effect of drought on seed traits in rice. So rice is among the top 10 crops cultivated worldwide, and is the staple food for more than half of the global population. Currently, rice production is already facing an emerging issue, which is water shortage. So in this plot here, I'm presenting the drought risk in the major rice cultivation area across the world. And you can see that some of them are already facing a serious water shortage. In particular, India and China, which are the two leading producing countries of the world. So for this reason, the rice breeders are getting more interested in developing drought-tolerant varieties. However, recent studies have posed another challenge, because they have shown that some of the genes that have been selected during the Green Revolution to have high-yielding individuals, they are tightly linked with QTL controlling drought tolerance in rice. Here there is an example from Vikram et al. So SD1 is the gene that is being extensively selected in the Green Revolution, and higher chance to develop seedlings under drought condition. In particular, seed weight and size depends on the grain filling process, which is divided in two phases: cell development, cell division, and cell elongation. And these two phases rely on a perfect balance between source and sink tissue. Of course, drought affects this balance because it decreases the photosynthetic efficiency of an individual, and it causes also it compromises the mobilization of nutrient from the source tissue to the sink. So somehow drought has many effects on influencing grain filling and therefore seed size and weight. So as I said before, seed size has been extensively studied in rice, but there is a lack of knowledge on how drought actually affects these traits. It is for two reasons. The first one is that it's kind of difficult and challenging to replicate big population under different environmental conditions, but at the same time, measuring seed size and weight is a very tedious work. But recently, the introduction of digital imaging has kind of facilitated this task. So we kind of solved the first limitation thanks to the development of new genetic material in rice at International Rice Research Institute. We chose to use the rice global magic population. So the magic population is population created with specific design where parents are intercrossed to each other. So in this way, you are kind of shuffling the genome, creating recombination, and then the possibility of finding markers with higher resolution. But the peculiarity of this population is that it combines two ecotypes of rice, indica and japonica. So indica individuals, they are grown mostly in tropical region, while japonica individuals are mostly found in temperate environments. And for the seed size, they present differences within and between subspecies. So we used 245 individuals of this population, and they were subjected to two environments. The first one is under seedling stage drought condition, and the second one is under flooded well water condition. In particular, we applied drought by stopping irrigation after 34 days from sowing, and then we resume irrigation at intervals afterward. And the second problem, as I say, is phenotyping, so collecting measurements of seed size and weight, in particular seed size. So in the traditional method is based on calipers, which is very time-consuming and laborious. We decided to apply digital imaging. In particular, we scan the seeds using a desktop scanner and a very simple setup, which include two color standards. They are essential for correcting differences in image exposure between images, and a black tray which confine the seeds in a specific space on the scanner. So we took images of more than 500 seed samples for an average of 110 seeds per sample. And we estimated that it took about 5 minutes to scan each sample. But then we had to process all these images, and we decided to use the open source Python package PlanCV, which has been released recently, and it includes a series of modular functions that you can use to extract every kind of information from plant images, including morphological measurements or color variation and so on. So we developed a pipeline using this tool, and this pipeline basically identify each seeds. This is why they have different colors here, and it estimates different parameters such as seed area, seed width, seed length, and so on. And it also has some measure of the shape of the seed. So we apply this pipeline to all images at the same time, And it took about 6 hours to process all seed scans on a common laptop. So if we consider the time we spent scanning the seeds and the time that we spent processing the images, it took about 1 month to get all the results, which is very fast and also accessible in terms of it's very cheap. But besides being rapid and cheap, our method was also able to capture the differences on these traits within our collection. So here you see that the diversity in the trait measure between the drought and well water samples. So the drought are the dots in purple, and the well water are the dots in orange. And you see that more than 60% of the phenotypic variation resides between these two groups. And they are different for specifically for almost all traits, but particularly for area, weight, perimeter, and major and minor axes, which are basically measure of seed length and weight respectively. This pipeline also provides measure of shape, such as ellipse, eccentricity. This is a measure of the roundness of the seed. So it is value from 0 to 1. And more close is to 1, more rounded is the seed. And the same is solidity is another measure of shape, seed shape. But of course, what we are interested in seeing if there are individuals that are drought tolerant under seedling stage. So in general, as expected, drought affect seed size and weight. In particular, we saw that most of the individuals produce smaller and shorter seeds under drought. But there are some samples that are kind of promising because they outperformed under drought or they maintain the same profile. So here I'm representing the trait values on the drought and well water condition. And the individual that are the dots that are above the diagonal indicate those individuals that are they have higher value for that trait under drought. So to better understand, if we look at seed area, which is a more intuitive trait, we see that here there are a few dots that are above the diagonal. So these individuals actually have higher area, seed area under drought condition, and could be potential donor of drought tolerance under seedling stage in rice. So this collection has been previously genotyped using genotyping by sequencing, and 2 different SNP dataset, variant dataset has been discovered using the Japonica and Indica reference genome respectively. And so we use this data to identify which genomic region are involved in controlling the traits that we measure. So here you can see that we have significant association, marked trait association in both environments. Some of these associations, they overlap in both environments, and some are environment specific. And so this is more clear here in this plot here. Basically, it's the same as before, but if you look from above. So here you have the dots that are basically the peak of that signals that you saw before. And you can see that some of the regions, as I say, they are shared in the 2 environments, and some we saw them some only in a specific environment. So these kind of indicate that the genetic architecture of seed-related traits change based on the environmental condition during rice growth. We saw that some of the non-genes controlling seed size are closer to the market trait association, such as grain size 5 and grain length 7. There is also this gene that causes incomplete grain filling, and this one is involved in starch synthesis in the seeds. So they can be good candidate for seed size. We also saw this region here on chromosome 12, and this locus is a non-major QTL controlling drought under reproductive stage. So the thing that we saw this signal also in seedling stage means that it has an involvement also under drought seedling stage. But the major conclusion of these results is that we suspect that G/E interaction have an important influence for seed size and the drought in rice. And so here they are more clear. So these are the reaction norm for the sites that we identify. So you have the effect of the measure allele on trait mean in the two environments, drought and well water environment. And you can see that the magnitude of the allele effect change based on the environment where the individual has been grown. So some of these alleles, some of these effects are conditionally neutral. So they have a high magnitude in one environment and no effect in the other. But we saw some antagonistic here for weight, which means that they cause an increase in seed weight under drought and a decrease of weight under well environment condition. And to make it even clear, we also saw that, so we identified this locus under the QTL controlling grain yield under reproductive drought stress. And we saw that this locus has a deletion of 50 base pair, and the individual that carry the deletion actually have a decreased value of eccentricity, which, as I say, is a measure of seed roundness. And this decrease is even more evident under drought stress. So altogether, these results tell us that G/E interaction have serious contributions to seed size variation under drought condition in rice, and they have to be accounted for during the selection of a drought-tolerant individual. It also so also this project also say that it was also possible thanks to digital imaging, which is a new promising area in agriculture, and also to close the genotype by genotype 3 phenotype gap. So as a conclusion of my entire talk, I'm referring to this book that I have read some time ago, which is a very interesting book, and it talks about the eternal dilemma between breeders and a part of breeders and a part of scientists, that they are saying, okay, we need to stop, we need to reinvent agriculture because of what is happening. I think based on my experience so far, I think plant breeding is resources that we can use, also based on the genomic tools that are now available, to kind of find a compromise between the two. And so we have kind of to reinvent agriculture to adapt to the future environmental scenario, and we have the instruments to do that. But this is the last slides. It was kind of difficult to list all the people that I have met in these 8 years. They have been a lot, and all of them have contributed. So this was possible thanks to the contribution of all of them. And I want to thank all of them because they contributed to my professional and personal development. And of course, thank you for listening.
Great. Can you hear me?
Yes.
I got disconnected in the middle there. Hello, everyone. Thank you so much, Annarita. You might want to stop sharing.
Yeah.
Okay, so the Q&A is open. It has been open the whole time, but please, you have one compliment. Nice presentation. Good job. But any questions for Annarita, please ask them in the Q&A. While we're waiting, I have a question. Mm-hmm. I think your last point is a really interesting one. This balance between how do we try and push forward agriculture using all these new technologies that kind of transforms the whole breeding program while choosing the right direction. Is that sort of the worry? I think I would like to hear a little bit more about how you think we can answer that as a community.
Well, I think it's a kind of still difficult, but so based on my experience with growers, it was kind of difficult to tell them, like for example, also Walnut is facing serious challenge with water. California is known to have serious drought problems. And we also worked with Iranian people where basically walnut tree, they grow almost in the desert. And so we were particularly interested to see how we can use those to bring tolerance, so we use less water. And so I think that, so what I say is that also it's also a responsibility of the geneticist and the plant breeder to teach growers to notice that they cannot do agriculture as they have been doing so far, and trying to keep like using these resources. And I think also, like in terms of science, if you like how I will do that, I think like I'm very interested, for example, in polygenic adaptation, the one that we are, because I think plants have a very, they are very useful to, like, kind of easy to study to understand how they already adapted to in the past to what happened. So we can use this information to see how to predict how they are going to do in the future. So that's kind of what I would like to work on now.
Sure. A question here: what is the approximate timeline in walnut breeding? How many years until an F1 can be evaluated for the desired traits phenotypically?
Oh, well, that's kind of I will say depend on the trait that you are interested in. So if you are considering, like, let's say, if you want to evaluate yield, you have to wait at least 5-6 years, 5-9 years. I think at 9 years that you have a stable production, meaning that, of course, when the tree starts to produce a walnut, it is not very, so it will start like slowly, first one, then the other, but you want to have a stable yield to be sure that it's a good candidate. So I will say 9 years, almost 9-10 years. But depends. If you are interested in root, that's gonna be easier.
Sure. Question: what might be the role of epigenetics in drought tolerance?
Oh, well, that's a very interesting question. It's also like something that really interests me, and I would like also to do that. I think it's a lot, like, I think epigenetics is another way to express G by E interaction somehow. So I think, yes, it has a main role, but it's also true that it's difficult to understand and to study epigenetics so far. But yeah, I think it has it has a role based on what we see just using like genetic markers and phenotype.
Okay, next question. For grape, is the conclusion that domesticated is more diverse than wild because the domesticated population that you used was created from hybridization among different wild collections? And also, was there more inbreeding in domesticated because they are hermaphrodites?
Oh, okay, that's a very good question. So the history of grapevine is kind of challenging because, so it's an outcrossing species and has many of outcrossing species. In general, they have high genetic diversity. But the level that we see today of genetic diversity is because of sexual crossing that human has been doing since the beginning, consciously and unconsciously, but also because, so when it was domesticated in the Near East, it was then brought to Europe. And while migrating, it was kind of introgressing variability from the wild. So yes, the wild population, they kind of contributed to what we see today. But I think it's also true that it is also say that in grapevine, the number of generations separating the whole vineyard to today, they are a few because the main agronomical practice was vegetative propagation. And so I think both phenomena, they have contributed. And then more inbreeding. No, the more inbreeding was in the wild one. So domesticated in general is very diverse. We didn't see inbreeding in the domesticated, at least in our data set. I think like inbreeding is more in the wild because it's the opposite. It's like you have male and female, so you have to have both individuals at the same time to have a cross. And since there is a small number of individuals, of course they are obliged to cross to each other. I think that's my interpretation.
Okay. Question: have you used genome prediction?
Oh no. Actually, that was the last goal in the Walnut breeding program, but my contract finished before that. But we are actually talking with the new plant walnut breeders, and we think that in walnut it's kind of be challenging doing genome prediction because we have seen allelic heterogeneity in the locus that we identified. And also it's very highly heterozygous. So defining, I think, the training population will be kind of difficult. But that's the ultimate goal. I think they are working on that now.
Okay. I'm going to ask one more question. Okay. So there's a question for a recommendation of approaches for genotyping. For example, how would you rate a SNP array over whole genome resequencing, etc.?
Oh, well, yeah, so I think it depends on the objective that you have. So I think now whole genome sequencing is very cheap. So if you have a very good reference genome, and you have money, you can do it instead of SNP array. The SNP array, they are useful if you have a common genetic basis on the population that you are screening. Because there is a kind of an ascertainment bias. So the SNP array in general, they are created because you do the resequencing of a panel, and then you find SNPs, and then you select which one to use in the SNP array. But of course, depends on which individual you are sequencing at the beginning. So if you have a common genetic basis, it's a good instrument, because it is also very good in genotyping heterozygous species compared, for example, to G bias. But genotyping by sequencing, I think it's better because it's cheaper, and it also like allows to test directly the diversity of your collection.
Okay, there's a number of other questions here, but we'll let Annarita maybe type the answer to those if she wants. Otherwise, thank you so much, Annarita, for your presentation. Thank all of you for your attention. Thanks for making this first seminar on the web and a successful one. And we'll be meeting, let's say, in about 5 minutes over at the Zoom happy hour. If you have that link, you're a member of the UMass Boston community, you're welcome to join. Bring snacks and beverages of your own if you want or not, and we'll have a more casual, regular sort of interaction there. Okay, thanks everyone. And Enrique, if you don't mind looking at the questions and if you want to answer them, go ahead.
Okay. Bye. Thank you. Bye-bye.